The treatment of certain cancers, of infections such as HIV or invasive fungal diseases, or of other serious medical conditions, relies on the regular administration of critical drugs, which means drugs that are absolutely required to keep those life-threatening diseases under control. Quite a number of these critical drugs are candidates for the monitoring of their blood concentration in the patients who receive them. Indeed, the concentration monitoring allows a precise personalization of treatments. This can improve long-term patient care, in particular for drugs with a highly variable and poorly predictable relationship between dose and concentration among individuals. This is particularly the case for drugs having a narrow therapeutic index and lacking clinically relevant biomarkers of efficacy and tolerability. And the aim of the IsSyPeM project is precisely to develop a new approach for therapeutic drug concentration monitoring at the point of care.
Donc ici, vous êtes au laboratoire de pharmacologie clinique.
Welcome to the Laboratory of Clinical Pharmacology. Our expertise is in the dosage of medication in the blood of patients. We are all different when it comes to medication. Some metabolize quickly, others more slowly, and we can optimize the tolerance profile and the efficiency of a medicine by adjusting the dosage according to the concentration of the blood.
En ajustant les posologies en fonction des concentrations sanguines.
At present, concentration measurements are performed in large medical laboratories using modern technologies, such as fluorescence polarization or liquid chromatography with mass spectrometry. The measurement results are sent to medical doctors in charge of the patients. However, the clinical interpretation of results represents a true challenge for non-expert practitioners. Indeed, it is fairly difficult to translate a mere drug concentration value into an appropriate decision of dosage adjustment in the patient. Clinical pharmacologists are experts in the field, but they are not largely available.
Therapeutic drug monitoring is currently limited because of a lack of software tools to help with the interpretation of drug concentration. One of the most critical and challenging goals of the IsSyPeM project was to design such a tool. And actually, within the last years, we achieved this goal. We now have a unique ergonomical software that is able to predict a future drug exposure of a specific patient and to propose a dosage adjustment to fit the specific needs. The software is now connected to the Lausanne University Hospital database and used by the pharmacologists in their daily practice. We are currently designing the clinical trials in order to make it a Class 1 medical device, and our startup is soon to be created with a close collaboration with the Lausanne University Hospital.
And more importantly, the IsSyPeM application is meant to be integrated with a point-of-care analytical system for the measurement of drug concentration in blood. This is also a part of the IsSyPeM project.
This system is able to measure the concentrations of drugs in blood samples directly, and it allows to display in real time the results.
L'avènement de petits appareils miniaturisés de ce type.
The arrival of miniaturized devices of this kind that are available to the medical practice of the polyclinic will allow us to democratize monitoring of the concentration of medication. Instead of having to send samples to big laboratories with the inevitable waiting list, the doctor will have the results directly on their screen. These will be immediately translated by an appropriate algorithm into the appropriate dosage for the patient, who will receive the doses of medication which are most likely to expose them to a concentration which is neither too strong and therefore toxic, nor too low and ineffective.
Thanks to the work of Professor Guiducci and to the personalization of medicinal treatments, electronics applied to the life sciences could, in the near future, avoid the discomfort of terrible side effects.
In the framework of the IsSyPeM Nanotera project, in the Laboratory of Microsystems of Professor Philippe Renaud, we have developed a novel blood sample preparation device. Blood microsample preparation is a major roadblock in most lab-on-chip approaches nowadays. They often require external machines or pipetting operations. Here we propose a novel passive on-chip method for extracting plasma from a whole blood that guarantees high purity and a large ratio of extracted volumes, starting from very small volumes. No external pumping system is required, as this system is capillary driven. Thus, the device is completely autonomous and very simple to use. The device yields plasma samples in a liquid form and could be integrated with many on-chip analytical systems or used with off-chip systems. The device has been developed in collaboration with DBS Systems, an analytics company in Geneva, and we are ready to patent for point-of-care blood microsample preparation.
Ultimately, the aim of the project is to provide the concentrational results to the prescribers and to be immediately exploited in order to support the dosage optimization of critical drugs.